Right, class. So I'm going to tell you some specific points over here related to the binding of this association of this carbohydrate with that protein. So this association of oligosaccharides, I can say repeating units of disaccharides, so you can say it is an oligosaccharides, right? So I told you already that they bind to the serine residue, and you must know and please know that obviously I'll be teaching about amino acid, but serine is a amino acid that is having a hydroxylic group attached with it. So this association of oligosaccharide with that OH group of serine, you can see over here. Look at this, right? So I'll be showing you out. Yeah. So when I'm talking about this, just a moment. Yeah, look at this. So this is association of serine. What happening? Yeah. So when serine is associating itself at this position, I mean from the OH or OH group of it, we call it as O-linked oligosaccharides, O-linked oligosaccharide chains. So it helps, it has role in recognition, interaction and enzyme regulation. Don't forget this. Recognition, interaction and enzyme regulation, and it binds with OH group of serine. Fine. Now, if you remember, N-acetylglucosamine is also a commonly O-linked, glycosylation of protein, serine. So it is a common, I mean you remember this N-acetylglucosamine. It is a commonly linked O-linked glycosylation. Many of the proteins, including enzyme factors, I mean certain enzyme transcription factors, are being regulated by this attachment, by this GLC-NAC attachment. We can say N-acetylglucosamine attachment. The attachment of GLC-NAC with this OH group um, is having certain regulatory effects, right? Stimulation or inhibition. So don't forget this O-linked association. Okay. Now N-linked oligosaccharides of glycoproteins are somewhat complex. You can see asparagine over here. This is serine or threonine, but look at ASN, that is asparagine. Now this is N-linked. If you remember, N-linked oligosaccharides is differing from O-linked. So what happened is that first the N-acetylglucosamine is linked to the protein via the side chain N of asparagine residue. You can see over here the side chain of asparagine residue. So this particular association needs to be remembered by you. In case of O-linked, it is serine or threonine. In case of N-linked, it is asparagine. Okay, so because of this, additional monosaccharides keeps on adding, and the N-linked oligosaccharide chain is modified by removal and addition of residue to yield a branch structure. So this is somewhat appearance happens. So NAN is N-acetylnuraminate, GAL is galactose. So just remember this structure, right? This is association of oligosaccharide with the serine or I can say the N-linked, I mean asparagine, right? So these are certain functions, certain associations of this. Remember the functions and all. Then I'll be describing lectins. So lectins are very important, and they are glycoproteins which recognize and bind to specific oligosaccharides. So in that case, the best example is concavalin A and wheat germ agglutinin. So they are plant lectins which are used as a research tools, right? So I'll be describing about lectins in my next lecture. Thank you.